Getting creatine into your muscles is relatively straightforward. Getting it into your brain is harder, slower, and governed by a completely different set of rules. Understanding those differences explains why the cognitive benefits of creatine take longer to appear than the physical ones, and why certain populations see dramatically stronger brain effects than others. Muscle uptake. Creatine is absorbed from your gut, enters circulation, and is transported into skeletal muscle via the SLC6A8 creatine transporter, a protein embedded in the muscle cell membrane. The transporter is abundant in muscle tissue and the process is efficient. Within 3 to 4 weeks of consistent supplementation, muscle creatine stores in most individuals reach full saturation. The brain is different at every step. Your brain is protected by the blood-brain barrier, a tightly regulated membrane formed by specialized endothelial cells lining the brain's capillaries. The blood-brain barrier controls what enters neural tissue with exceptional precision. Creatine is not freely permeable across it. It requires a dedicated transporter, the CRT1 protein, located on the blood-brain barrier endothelial cells, to cross from blood into brain. This transport is slower and more limited in capacity than the transporter in muscle tissue. As a result, brain creatine levels rise with supplementation, but substantially more slowly than muscle creatine. Magnetic resonance spectroscopy studies, a non-invasive imaging technique that can detect and quantify brain metabolite concentrations, show that meaningful elevation of brain creatine typically requires 6 to 8 weeks of consistent supplementation. Compare that to 3 to 4 weeks for muscle. This delay has practical implications. If you're supplementing primarily for cognitive benefits, and you stop at 4 weeks because you haven't noticed a difference in mental performance, you may be quitting before your brain creatine has had time to rise meaningfully. The brain also synthesizes some of its own creatine locally. Both AGAT and GAMT, the enzymes responsible for creatine synthesis, are expressed in certain brain cell types, including astrocytes. This local synthesis partially compensates for restricted transport across the blood-brain barrier, but it's not unlimited. Individuals with mutations in AGAT or GAMT develop severe neurological disorders despite having intact transport systems, proving local synthesis alone cannot meet the brain's full creatine requirement. Why does brain creatine matter so much? Neurons fire action potentials by rapidly moving ions across membranes using ATP-consuming pumps. This happens billions of times per second across the entire brain. The brain accounts for just 2% of body weight, but consumes roughly 20% of the body's resting energy. During intense cognitive tasks, local ATP demand in active brain regions can spike sharply. Phosphocreatine in neurons acts as the same instantaneous ATP buffer it does in muscle, rapidly regenerating ATP when local demand exceeds local supply. Higher brain creatine means a larger buffer against those demand spikes, which translates to sustained cognitive performance under load. The populations showing the largest cognitive response to creatine are exactly those with the lowest baseline brain creatine: vegetarians, vegans, and individuals under chronic stress or sleep deprivation. In clinical trials on sleep-deprived individuals, creatine has been shown to significantly blunt the cognitive decline that normally accompanies prolonged wakefulness. In student populations during exam periods, supplementation has shown improvements in processing speed and working memory compared to placebo. In some studies, the magnitude of cognitive improvement in vegetarians rivals the effect size seen for physical performance improvements in strength athletes. For anyone whose primary reason for taking creatine is brain performance, the protocol is the same as for physical performance: 3 to 5 grams per day, taken consistently. The only real difference is patience. Give it 8 weeks before evaluating, because the tissue you're trying to load has a slower, more regulated uptake mechanism than the one in your quads. There's another practical implication of the brain's slower creatine kinetics. Single high-dose strategies don't work the way they do for muscle. Loading 20 grams per day for a week floods muscle stores efficiently because muscle transporters can handle the rapid influx. The blood-brain barrier's slower transport mechanism means that large short-term doses don't accelerate brain creatine loading the way they accelerate muscle loading. For the brain, the most effective approach is a consistent moderate dose maintained over an extended period, not a loading phase followed by maintenance.